From scattering them in salads to slicing them in sandwiches, the tomato is a picnic time favorite, as long as it's not soft. But the precise mechanisms involved in tomato softening have remained a mystery until now. Biologists have identified a gene that encodes an enzyme which plays a crucial role in controlling softening of the tomato, which could lead to new varieties of better-tasting fruit that stay firmer for longer. The tomato is one of the most valuable fruit crops in the world with an annual global value in excess of $50 billion. Consequently, plant breeders are working continuously to supply high-yielding, better-tasting, more nutritious and longer-lasting tomato varieties, but some of the best-tasting varieties soften rapidly and can have a short shelf life. To support the tomato industry and further improve consumer satisfaction with new tomato varieties, a major scientific goal has been to identify genes that allow the targeted control of fruit softening without impacting other aspects of ripening, said lead author Professor Graham Seymour, of the School of Biosciences at the University of Nottingham. Such work would permit excellent fruit flavor and color development, combined with enhanced shelf life. To reduce wastage, hybrid tomatoes are often bred to include natural mutations that slow the whole ripening process. But such longevity comes at a cost, in the form of flavor and color. The question of how the tomato fruit disassembles its cell walls and softens during ripening has perplexed researchers for over 2 decades, but the study, published in the journal Nature Biotechnology, has revealed the key to uncoupling softening from the other aspects of fruit quality. Together with scientists from the Royal Holloway, University of London, the researchers have identified a gene that encodes a pectate lyase, which normally degrades the pectin in the tomato cell walls during ripening. Professor Seymour said, "In laboratory experiments we have demonstrated that if this gene is turned off, the fruit soften much more slowly, but still show normal changes in color and the accumulation of taste compounds such as acids, sugars and aroma volatiles. Natural variation exists in the levels of pectate lyase gene expression in wild relatives of cultivated tomato, and these can be used for conventional breeding purposes. This discovery can provide a means to refine the control of fruit softening in modern tomato cultivars." Dr. Charles Baxter from agrochemical company Syngenta said, "This discovery has relevance for the development of new tomato varieties via conventional plant breeding and is a significant step forward in understanding processes involved in fruit development, allowing more refined control of this process in plant breeding. Using conventional plant breeding, tomato wild species can be readily crossed with a cultivated tomato. One wild species, Solanum penlei, has low levels of pectate lyase gene expression in the fruit. This genetic variation can be used to breed slow-softening cultivated tomatoes." Paul Fraser, professor of biochemistry at Royal Holloway said, "The study also shows how you can precisely alter fruit ripening properties without adverse effects on the chemical composition of the fruit. In this way the consumer traits such as taste, color, and nutritional quality are not adversely affected and in some cases enhanced."